Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine an AI that doesn't just find new drugs, but actually invents them from scratch, creating molecules structurally unlike anything we've ever used before.
Yeah, it sounds like science fiction almost.
Right. But that's exactly what a groundbreaking paper in Cell from 2025 has just achieved.
Yeah.
Generative AI inventing antibiotics way beyond the known chemical structures. It's like, I don't know, discovering a whole new musical scale for medicine.
It really is a pivotal moment, and our mission today really is to give you a powerful shortcut to being well-informed about this. It's a critical breakthrough.
Okay.
We'll unpack how this artificial intelligence is opening up entirely new frontiers in the fight against, superbugs, helping you understand not just what they did, but maybe more importantly, why it matters so profoundly for our future.
All right, let's dive in. And maybe we should start with the core problem first, because it's urgent.
Absolutely urgent.
We are facing this truly dire and, well, escalating crisis, antimicrobial resistance, AMR. And this isn't some, far-off threat. It's happening right now, globally. Antibiotic resistance is just outpacing our discovery of new drugs.
It really is. We're talking about approximately 5 million deaths annually linked to AMR.
5 million. That number is just staggering.
It's hard to even grasp. Think about it. That's roughly the entire population of, say, Ireland or New Zealand dying every single year just from infections we can no longer reliably treat.
And it's growing.
It is. And that grim reality is made worse by the fact that our traditional antibiotic discovery pipeline, well, it's thinned out dramatically.
Why is that? What happened to the pipeline?
Think of it like this. Our existing chemical libraries, those huge collections of molecules scientists have built up over decades, they've essentially been mined repeatedly.
Okay, we've looked through them already.
Over and over. And the conventional screening methods, they just aren't yielding truly novel structures anymore. Not the ones we need.
What makes a structure novel in this context?
Well, we're looking for compounds that can balance effectiveness against the bacteria, crucially selectivity, so they don't harm our own human cells, and they need the right drug-like properties to actually work inside the body.
Right. It's not enough for it to just kill bacteria in a dish.
Exactly. Finding that perfect combination with the old methods has become incredibly difficult, so we're left desperately needing something new, something structurally novel.
So we're kind of stuck, old tools getting rusty, and we need to break free from just tweaking what we already know.
And that's precisely where this AI promise comes in. It's a potential way out.
Okay, so tell us about that. How does AI help here?
Well, what's fascinating, the core idea of this paper, is that modern generative AI models can actually overcome these limitations. Instead of just searching existing compounds like sifting through a pre-existing pile of Lego bricks.
Yeah, I like that analogy.
They can propose entirely new molecules from scratch. Design them. These AI systems can design molecules optimized across multiple goals at the same time. Strong activity against the bugs, high selectivity for safety, and really importantly, synthesizability.
Synthesizability meaning they can actually be made.
Exactly. Can they actually be built in a lab, not just exist as a cool idea on a computer screen?
So the AI designs these potential drugs in silico in the computer simulation before anyone commits to expensive, slow lab work.
Precisely. It's a kind of full stack approach, you could say. Machine learning generates the ideas, then those ideas get passed to the lab for synthesis, and finally they go through rigorous testing and mechanistic profiling, figuring out how they work.
A real end-to-end pipeline, trying to speed things up dramatically.
That's the goal.
Right, so how did they actually build this groundbreaking pipeline? Where did they start?
Well, the journey starts with something that might seem small, but it's fundamental. Screening what they call chemical fragments.
Fragments? Okay, like little pieces of molecules.
Exactly. Phase 1 was all about screening these chemical fragments. Think of them as the basic, often simple building blocks of larger molecules.
And why start there? Why not jump straight to full molecules?
Good question. It's because these fragments often hold the key to a compound's core biological activity. It's like finding a really powerful engine part before you design the whole car around it.
Okay, got it. And AI is powering this search.
Oh, absolutely. They didn't just randomly pick fragments. They used graph neural networks, GNNs, a type of AI that's particularly good at understanding complex relationships within structures, like how atoms connect in a molecule.
Right, suited for chemistry.
Very much so. And these GNNs were used to intelligently screen over 45 million chemical fragments.
45 million.
It's an enormous number. Imagine trying to manually screen that many unique grains of sand to find the perfect ones. AI basically transformed that impossible task into something manageable.
Incredible scale. But I guess it's not just about the number, right? It's about finding the right ones.
Precisely. The key isn't just volume. It's the smart filtering process they applied. They meticulously filtered these millions down to identify only the most promising candidates.
How did they filter? What were the criteria?
Several crucial steps. First, predicting antibacterial activity against really urgent priority pathogens, like Neisseria gonorrhoeae that causes gonorrhea, or Staphylococcus aureus, staph infections.
Okay, so activity first. But what about safety?
Critical point. They also had to make sure these fragments weren't toxic to human cells. So they removed any fragments predicted to harm various human cell lines.
Makes sense.
And crucially, they excluded fragments with these things called panes and brink substructures.
Panes and brink. Sounds ominous.
They're basically chemical red flags, patterns in molecules known to cause problems, like binding randomly to too many things in the body, which leads to side effects, or just makes the drug useless.
Okay, so weed out the troublemakers early.
Exactly. And here's where the novelty part really comes in strong. They enforced structural distinctiveness. They removed fragments that were too similar to any known antibiotics.
How similar is too similar?
They used a specific metric, Tanimoto similarity less than 0.5, to ensure a significant structural difference from anything we've seen before. It's making sure the new molecule isn't just a slightly tweaked version of an old one, but really charting new chemical territory.
Right, truly novel. So after all that intense filtering, what was the payoff from this first phase?
The payoff was huge. This rigorous screening identified over 1 million unique fragments. And what's truly fascinating is that many of these expanded way beyond the known chemical diversity of existing antibiotics.
You could see that in the data.
Yeah, they had these visualizations, TSNE plots, showing these new fragments clustering in previously unexplored chemical spaces. It really proved their models could reliably find promising, novel starting points.
Okay, so they had these promising fragments. What's next? Building the car from the engine part.
Exactly. They were ready for the next challenge, building full molecules from those tiny beginnings. This is phase 2, where the generative design truly kicks in.
This is the exciting part, right? The invention part.
Definitely. They took those carefully selected fragments and used 2 different generative AI algorithms to expand them into entirely new, full-size molecules.
2 different AIs.
Yes, one was a genetic algorithm called CREM. It sort of evolves molecules through chemically reasonable changes. And the other was a fragment-based variational autoencoder, an FVAE, which learns how to generate new molecules by combining fragments it's seen.
So different approaches to building molecules.
Yeah.
And did they just spit out possibilities?
Not randomly. It was an iterative refinement process, a kind of closed learning loop. They generated millions of new compounds, but constantly improved them through rounds of selection.
Selection based on what?
Based on the AI's predictions for, again, antibiotic activity, low toxicity to human cells, and importantly, how easy they'd likely be to synthesize in the lab.
That synthesizability again. Practicality matters.
Always. It's a continuous feedback cycle where the AI gets smarter with every round, hopefully homing in on better and better candidates.
And did it work? What was the first big success story from this phase?
The compound they named NG1. Get this. Out of just 24 computationally designed and custom synthesized compounds, which is a tiny number in drug discovery, 7 showed selective antibacterial activity.
7 out of 24. It's pretty good.
It's excellent. And NG1, designed using that FVAE model, was a real standout.
What was so special about NG1?
Remarkable potency. Its MIC, that's the minimum inhibitory concentration, basically how little drug you need, was just 0.5 micrograms per milliliter against N. gonorrhoeae. very potent.
And selective, you said.
Yes, narrow-spectrum activity. It specifically targeted the pathogenic Neisseria species, while, and this is crucial, sparing the commensal, the beneficial bacteria in our bodies.
That's a huge advantage. Broad-spectrum antibiotics can really mess up your gut microbiome.
Exactly. And tackling resistance. NG1 proved effective against highly drug-resistant and gonorrhoeae strains, including those resistant to multiple existing treatments.
Wow. So it's not just new, it hits the really tough bugs.
Right. But here's where it gets really, really interesting. Its unique mechanism of action, how it actually works.
Okay, tell me.
So NG1 was found to be bactericidal. It actively kills the bacteria, doesn't just inhibit them, mostly by damaging their membranes. But fascinatingly, it also targets a specific protein called LPTA.
LPTA. Never heard of it.
Most haven't in this context. It's a critical part of the lipooligosaccharide, or LOS, export system. Basically, a system the bacteria need to build and maintain their outer protective layer.
So wait, LPTA is a novel target, an undrugged target.
Precisely. An undrugged antibiotic target in the LOS biosynthesis pathway. That's the real breakthrough here, arguably.
Because we're not just finding new keys for old locks.
Exactly. AI is helping design keys for entirely new locks that bacteria haven't encountered before, haven't evolved resistance to yet. This could be critical for staying ahead.
Mind blown. Okay. And did it look promising beyond the lab dish, like safety? Animal models?
Yes, good signs there too. Positive toxicology studies, it wasn't damaging red blood cells, wasn't causing mutations, and importantly, it showed in vivo efficacy.
In live animals?
Yes, in a mouse model of gonorrhoeae vaginal infection, it significantly reduced the bacterial load. That demonstrates real potential to move towards actual clinical applications.
Incredible. So NG1 is a huge success story for Neisseria. Did they stop there or try this approach on other bugs?
They kept going. Which leads us into their phase 3. They asked, can we apply the same powerful approach to other pathogens? And can we push the AI even further?
Okay, what other pathogens?
They turned to gram-positive bacteria, specifically targeting Staphylococcus aureus, including the really problematic MRSA methicillin-resistant Staph aureus.
The hospital superbug.
That's the one. Using the same fragment-based approach, they identified another promising fragment, F2, and designed a compound called EN1, which showed activity against MRSA.
Another hit. But you mentioned synthesizability earlier.
Yeah.
Was making EN1 or other compounds like it still a bottleneck?
Ah, yes. And this is a really important dose of realism in drug discovery, even with AI. Synthesizability, how feasible and economical it is to actually make these molecules, is a huge factor.
So the AI can dream up amazing structures, but making them is another story.
Sometimes, yes. Many novel structures designed by AI can be incredibly complex. Imagine designing a totally new kind of intricate sculpture that needs special tools and materials that barely exist.
Right?
Synthesizing some AI molecules can be like that. There's often this trade-off. A molecule might look amazing on the computer, fantastic predicted activity, but be incredibly hard or expensive to actually produce in the lab.
So AI is powerful, but it still needs human chemists to figure out the how to make it part.
Absolutely. It highlights that AI is a design tool, a powerful one, but it works best hand in hand with expert chemists to bring these digital ideas into physical reality. It's a collaboration.
That makes perfect sense. Balancing ambition with practicality. Okay, so after tackling staph, what was the next big leap?
Perhaps the most audacious step. Unconstrained invention. De novo design.
De novo. Learning from the beginning.
Exactly. Designing antibiotics without any specific fragment as a starting point. It's like telling the AI, forget the Lego instructions, forget the known parts, just build me something amazing from raw plastic.
Wow. How do you even do that?
They use models like the junction tree variational autoencoder, or JTVAE. These essentially learn the underlying rules of chemistry and molecule building and then generate completely novel structures from scratch.
And did that work? Seems like a long shot.
Amazingly, yes. They had more success stories, compounds named DN1 through DN6, out of just 22 de novo synthesized compounds again, synthesized from pure AI invention. An impressive 6 showed antibacterial activity.
6 out of 22 from scratch. That discovery rate sounds incredibly high for completely new chemical space.
It really is remarkable, and among these, DN1 really stood out.
What was special about DN1?
It showed broad spectrum activity. It hit gram-positive bacteria, including multidrug-resistant MRSA, but also, surprisingly, N. gonorrhoeae, too.
Whoa, broad spectrum from a de novo design. That's valuable.
Very valuable. And what's fascinating about DN1 is its distinct mechanism. Like NG1, it's membrane active. It messes with the bacterial cell membrane.
How could they tell?
They could actually visualize the damage using cryo-TEM images, this super high-res microscopy. You could literally see things like the membrane thickening up in S. aureus or getting dislodged and swelling in N. gonorrhoeae.
Visual proof of the damage.
Exactly. And it highlights that even structurally diverse compounds cooked up by AI can have distinct yet highly effective ways of attacking that bacterial fortress, often hitting the membrane.
And what about resistance? And animal studies for DN1.
Crucially, DN1 showed a low frequency of resistance development. That's fantastic news for long-term use. Plus, rapid bactericidal activity, it kills bacteria quickly, even faster than vancomycin against MRSA in their tests.
Faster than vancomycin. That's a major benchmark.
It is. And just like NG1, DN1 significantly reduced bacterial load in a relevant animal model this time, a mouse model of MRSA skin infection. It performed similarly to a standard clinical antibiotic control.
Another big step towards real-world translation.
Absolutely. Bench-to-mouse validation is key.
Okay, this whole deep dive, it really feels like it showcases a new era. What makes this work, this paper, a true breakthrough, beyond just, cool science?
Well, first and critically, it moves far beyond simple rediscovery. This isn't just screening old libraries slightly better or re-ranking known compounds. It genuinely explores uncharted chemical space.
Creating truly structurally distinct antibiotics.
Precisely, which means bacteria are much less likely to have pre-existing resistance mechanisms ready for them. It's truly expanding our chemical arsenal in a novel way.
And it's not just theory, as you said. The bench-to-mouse validation is key.
Absolutely critical. Showing efficacy not just in a petri dish, but in vivo in animal models. That bridges the gap between a computational prediction and a real biological effect. It proves these AI molecules aren't just, pretty pictures.
Right.
Furthermore, they establish that closed learning loop. Remember how the feedback from the lab tests updated the AI models?
Yeah, the AI learns from its own experiments.
Exactly. It creates this iterative, data-efficient discovery loop for continuous improvement. The AI gets smarter and more accurate with every single experiment. It's learning on the job.
And you mentioned collaboration earlier. This wasn't just AI gurus or lab biologists alone.
Not at all. This success is really a prototypical AI wet lab fusion. It absolutely required seamless, genuine collaboration between the machine learning scientists, the synthetic chemists who could actually make these new molecules,
The microbiologists testing them.
the pharmacologists running the animal studies. It highlights the intensely interdisciplinary, team-based environment you need for these kinds of monumental breakthroughs. Everyone has to speak the same language or learn to.
So, thinking bigger picture then, what does this mean for the future of antibiotic discovery?
Well, I think this platform offers nothing less than a potential template for an antibiotic discovery renaissance, one that's anchored in AI-guided exploration of chemical space.
A renaissance. That's a strong word.
I think it's justified. And it's an extensible platform. The same fundamental approach could be applied to other pathogens, other mechanisms of action, maybe even other types of drugs beyond antibiotics.
So what are the next steps? Where does the research go from here?
The future directions are also really exciting. They point towards using multi-objective optimization frameworks. Basically, teaching the AI to simultaneously find compounds that tick multiple critical boxes at once. High antibacterial activity, good synthesizability, easy to make low toxicity, and other favorable drug-like properties, like a metabolic stability, making sure it lasts long enough in the body.
So asking the AI to find the perfect candidate that meets all the criteria.
Pretty much. Like asking it to find the perfect house that's beautiful, affordable, well-located, big yard, good schools, all at once. Trying to optimize everything simultaneously.
But there are still challenges, right?
Yeah.
You mentioned synthesis throughput.
Yes, and that's a key acknowledgement. Making these amazing AI-designed molecules quickly and cheaply is still a major hurdle. Synthesis is often the bottleneck.
So we need better ways to predict synthesizability and better ways to figure out how to make complex molecules.
Exactly. We need ongoing improvements in those synthesizability predictors and also in complex retrosynthesis algorithms, AI that helps figure out the reaction steps needed. That's crucial to unlock even more of these AI designs for real-world testing and use. That's where a lot of innovation still needs to happen.
Okay. So wrapping up, this deep dive has really shown us that AI isn't just like helping out anymore. It's actively inventing in this incredibly crucial field of antibiotic discovery.
Yeah, it's providing a credible path forward, a way to potentially expand our chemical arsenal against these increasingly resistant pathogens. It's giving us genuinely new weapons in an escalating war.
It really feels like a game changer, moving beyond finding needles in haystacks to, as you said, growing completely new haystacks full of unique needles.
That's a good way to put it.
So a final thought to leave our listeners with. As this AI continues to learn, evolve, get better, and as our ability to synthesize what it designs also improves, what other seemingly impossible medical challenges could it help us overcome? Could this maybe be the beginning of the end for the superbug crisis? Or perhaps at least a powerful new way to finally keep pace with it?